Hello, everybody, and welcome to the SCHOTT Pharma podcast. Today's topic is how to choose the right vial for next generation therapies. To learn more about the challenges that new drugs bring with them and how the criteria for taking this right selection to safely pack them, I have with me Diana Löber. Diana, maybe well known to some of you, is Global Product Manager at SCHOTT Pharma and is responsible for specialty and sterile vials. Diana leads and coordinates developments for the glass vials world. And as I like to say, Diana is our vial champion. Hi, Diana. Nice to have you here today again.
Hi, Niels. Thanks for the nice introduction. Nice to be here again.
Okay, so let's start, Diana. My first question would be, so as said, your Pharmaversity session is called How to Choose the Right Vial for Next Generation Therapies. But first of all, what exactly do you mean or do you understand behind next generation therapies?
So, during the last decades, the term biologics has been well established. What is often not clear at the first sight is the overwhelming diversity and complexity behind. So, by nature, these are typically therapies that cannot be pushed into one category. There is no one fits all approach possible. And in the end, every therapy, every molecule is unique, resulting in specific requirements. And based on this uniqueness, there is a huge diversity of requirements, what is especially valid for the primary packaging. And this is why I didn't refer to biologics here to avoid the impression that this is only one category. So there are actually many categories behind and many new applications possible. And therefore, I use the term next generation therapies. So implicating that there is still something next to come.
Understood. So if we look well back and ahead, so what has been already established somehow, and what is this yet to come you were mentioning?
So it all started with genetic engineering. So the ability to create artificial genes in the lab, and that was in the 70s. And this provided the basis for the development of biologic drugs. And the first monoclonal antibody has been generated in 1975. But the first FDA approved monoclonal antibody was orthoclone, and this was in 1986. And it was for a transplant, or it was a transplant rejection drug. And I think today we are somewhere between 150 and 200 approved MAPs. And since the COVID pandemic, DNA and mRNA vaccines are state of the art. So it was amazing to see how this accelerated the development here. And even though cell and gene therapies are complex from multiple angles, they gain more and more approvals year over year as well. And with these applications, the end of the line has definitely not been reached yet. And for those who listen carefully to my last Pharmaversity, so ADCs and RDCs or RACs are finally on the rise as well. And also for these applications, more is possible. There is already work being done on so-called iADCs. So these are molecules that combine tumor-targeted cell killing with immune activation. Or there is ADCCs, so that basically speaking, use a monoclonal antibody to bring a specific immune cell to the cancer cell so that they then do the job, let's say.
Well, amazing. So I understand great achievements have been realized and much more is coming. You mentioned seconds ago that these diverse applications also lead to diversity in the requirements for the primary packaging. Could you mention some of these requirements? What exactly do you understand?
So one thing that we typically see is that 50 to 60% of the applications in this segment, they need lyophilization. And this is a really steady number, a really steady share that simply does not change over the years. And due to the fact that freeze-drying is very complicated in syringes or cartridges, this is an application that simply stays in vials. This is a complex process, and the wrong container choice can significantly increase reject rates and therefore costs in the end. And in the last years, we have for the first time also seen the significant rise of therapies that require cold storage. So not only fridge temperature, but frozen, sometimes even deep cold storage, so down to minus 80, 120, even further. And this is always the case when you have a certain sensitivity, as for example, cells or viral vectors have. The risk for mutation is simply too high if they are in liquid. And also in lyo, they cannot survive so far.
Or maybe here I'd like to add for everyone who like to know more about that. So we had a previous Pharmaversity session on that, specifically vials for deep cold storage.
Exactly. Thanks, Niels, for mentioning this. What we've also seen, or what we also see, is a higher focus on efficiency. And this topic especially gained relevance during the pandemic. Yeah, so there it was about increasing speed to faster produce huge quantities, to fill huge quantities of vials. But also the trend towards small batches needs different consideration when it comes to efficiency. So how can I quickly perform changeovers? How can I keep the needed footprint as small as possible? But based on my experience, the most burning questions we get from customers are related to drug container interactions for liquid fills. And for that, vials are still the most prominent container format.
Okay, and why is it actually, Diana? So I would think that, well, putting it directly into a syringe would make the whole thing much more sense and easier.
Yeah, so syringes are of course very easy to use, but a lot of different components or materials are in contact with the drug, and this increases complexity because a multitude of possible interactions exist. You have the silicone, the needle, the cap, the plunger, and of course the glass. And when using a vial, only the glass is in constant direct contact with the drug. The influence of the rubber stopper is less prominent, and you have medium level of ease of use. And of course, the variability of dedicated surfaces you can choose from is the biggest for vials.
Okay. So then if I would need to draw a conclusion for me, so would it be correct to say for biologics, probably you need lyophilization, and so take a vial. For liquids, in case of very sensitive drugs that need specific container surfaces, take a vial too.
Yeah, so actually you could summarize it like that. Of course, in the end, also costs play a role, though. But I mean, this is something we are not going to evaluate in this Pharmaversity.
Oh, no, thanks, you're right. This not the focus today. So coming back to the topic, you mentioned liquid fill as the most burning topic. What is going on here?
So perfect introduction, Neos, because the most is going on here. To be more specific, the most drug container interaction. This is, of course, the most reactive combination. No lyo, no freezing, a liquid constantly in contact with the inner wall of the primary packaging.
Yeah, and this reminds me of chemical lessons a while ago.
Keep that in mind because there is more to come that you might still remember.
Okay, I'm intrigued. Let's go.
So let's come to the first influence factor. And by the way, in total, we will cover 6. Yeah. So just as a spoiler. And the first influence factor is filling volume. And there, especially critical is a low fill volume. So why is that? Of course, the lower the filling volume, the higher is the surface area in relation to that filling volume. What is, of course, the reason? The bottom. The more you fill, the more negligible the influence of the bottom gets. And now there are actually 2 topics connected with that. So the first topic is, especially when talking about high value drugs that are, by the way, often very low filled, every droplet counts. And some molecules, respectively formulations, tend to stick to the surface. And some molecules adsorb to the surface in that way that even mechanical impact, so aka shaking, does not help. And consequence then is that the concentration is lowered. It is not ensured that the drug gets injected to the patient in the right dosage. The second topic related to filling volume is something that is strongly connected with the vial forming process. So during that process, you heat the glass tube, and with that evaporates occur that recondense on the nearest cooler region of the vial's interior surface, so the so-called heel zone. And that is a zone just above the heel of the vial, so close near to the bottom area. And this area is less chemically durable. It shows a higher leaching and is therefore more susceptible to the processes that lead to delamination. So now really imagine a low fill volume vial before your inner eye. Of course, the higher leachable level of that heel zone has a much higher impact for a lower volume. As a consequence, the leachables per filled milliliter are much more elevated for a 2R vial filled with 0.5 milliliters compared to a 2R vial filled with 2 milliliters. So the first learning is filling volume is not just something that needs to be considered during the filling process. It needs to be taken into consideration much earlier during the development.
Okay, understood. And great explanations, Ayan. I think it's pretty clear. So that was number one. What about the next factor of importance?
So the second influencing factor I would like to mention is pH. And typically with regards to pH, you try to stay in the range of pH 5 to 7, so more or less in this neutral area, or sometimes also called physiological pH area. And this, of course, plays a role when it comes to the experience, to the sensation of pain for the patient. Yeah. But what we have seen for quite a long time now is that there are sometimes also drugs that use buffers or have to use buffers in the acidic range. Yeah. And a quite new trend is that for recent years, we have also noticed more and more drugs are also buffered in the alkaline range. So sometimes up to a pH of almost 11. Yeah. But extreme pH areas both are a challenge for standard borosilicate glass vials. So in low pH, the so-called hydrogen ion attack takes place. So a mechanism in which an ion exchange of positive hydrogen ions with network modifiers of the glass, for example, sodium, takes place. And this leads to a rise of pH. So, and in the end, the stability of the formulation is endangered. When it comes to a high pH, a hydroxide ion attack takes place. This attack is continuous and dissolutes the silicon-oxygen bonds of the glass network, releasing network modifiers elements into the solution. And these leachables might have an impact on the drug, resulting in denaturation of the protein, aggregation, or even deactivation. And this is, of course, something that needs to be very carefully evaluated.
Okay, so I've taken filling volume, key relevant, understanding of pH behavior, key relevant. So now I'm on fire, Diana. What is next?
So, Niels, you must have felt it. So, the next topic is temperature. And this is why I mentioned in the beginning that you might be reminded of the chemical lessons at school. So, temperature is simply an accelerator when it comes to chemical reactions. And with regards to temperature, actually, there are two topics concerned. Yeah, so one is terminal sterilization, and the second is storage temperature. So regarding the first one, terminal sterilization, I think it is very easy to imagine how terminal sterilization affects the drug container interaction. So basically, you cook it. And why do we, for example, put a laurel leaf in our stew when cooking? We want to extract the taste. And before my inner eye, something similar happens here. So the extraction of the glass leachables, even though you don't want it. Yeah, with the laurel leaf, of course, you want it. So the second one, so the storage temperature. So what we usually do to evaluate shelf life is what we call accelerated aging. So, and what do we do here? We increase the ambient temperature to stimulate, to mimic a certain amount of shelf life. And nowadays we have gained a very good understanding of what storage period in combination with what temperature equals what real life storage time. Of course, when it comes to the stability of the drug, the ambient temperature during storage needs to be taken into consideration.
That totally makes sense. Okay, so if I counted correctly, we've covered now 4 out of 6 factors or key factors. So I'm very excited about the fifth one.
So actually, that is something we implicitly already talked about, but what needs to be additionally highlighted, leachables. I mentioned this already with regard to the impact for low fill volume, but there is more to it. There are simply APIs that are sensitive towards glass leachables. And it is not black or white or yes or no, but what we can state is that with the increasing complexity of the molecule increases the risk for sensitivity towards leachables. And what can happen is that proteins denature, unfold, agglomerate, bearing the risk that the protein loses its activity, making the drug ineffective.
Which would be very bad, right? So apparently something that needs to be very evaluated carefully, no? Because I understand it is really unique for each drug. Okay, so this was the fifth, and we're coming to the last one.
So last but not least is definitely the age of the drug. So what do I mean by that? So in which development stage is the drug? Why do I mention this? Of course, there are commercial drugs that would be better off with a different packaging, but the hurdles to change that, to touch the registration and change the primary packaging are, of course, huge. So based on our experience, our customers only do that if there has been a warning by a regulatory agency. So if there is a real danger for patients' lives. And the good news is that there are things you can do without touching the registration.
Okay, this sounds interesting. So we've heard now from you the 6 influencing factors so that we can choose the right container for the liquid fill. We have the filling volume, the pH, the terminal sterilization, storage temperature, leachable sensitivity, and as you called it, the age of the drug. This is quite a lot.
Indeed. But this is honestly what I wanted to achieve, to show the complexity, to show how these different requirements need to be considered when choosing your primary packaging.
Okay, but so far we have not yet chosen anything.
That's true. But this is why SCHOTT Pharma exists and why we have such an elaborated portfolio that tackles every challenge.
Okay, so what do you have here to offer?
So as you can imagine, the combination of factors is somehow endless. But I will try to pick out some examples that gives you and of course our listeners an impression here. So first case is what I will call the modest traditional drug. So a filling volume that is close to nominal, let's say 2 milliliters in a 2-hour vial, in neutral pH, no terminal sterilization. And let's even assume that it isn't stored by fridge temperature. You might now think, here a standard conventional inner surface is fine. And it could be. Now the but. Even for that combination of factors, it is heavily recommended to perform screening studies according to USP 1660. This is only a recommendation chapter, but it helps you to rule out an elevated risk for delamination.
So, what if this screening study would show, let's say, no additional risk? Would it be fine to still use a conventional vial?
In general, yes, but especially for biologics, we recommend to use every pure vials. I do not deny that we want to sell our products, yeah, but for this specific one, it is really a matter of my deep conviction. It is difficult for me to understand why not simply choose the best type 1 glass inner surface, especially for high value drugs, or for diluents that are used for high value drugs, if there is one existing. And to say that in advance, yes, it has a higher price, but I can state that this is reasonable, yeah, and in a reasonable range, and not X times of a conventional vial.
By the way, there's a session on that too.
Sure.
So choose the right vial from the very beginning. Okay, so coming back here to our topic. So what requirements would lead to the usage of a vial with another inner surface then?
Yeah, so imagine you have a monoclonal antibody, very large, complex molecule. I would call this case like the default biologic. Of course, as typical for biologics, a low filling volume and with a pH, let's say, in neutral range. You might think, yeah, everything is fine, but you definitely should check if this specific molecule is sensitive towards glass leachables like sodium, aluminum, boron, calcium. It cannot be avoided that there are glass leachables. And you have to make sure that your drug is stable in the end. And there, type 1 plus would be the right inner surface. And if you had high pH, all alarm bells should go off there. You should definitely test every care.
So talking about these inner surfaces, who were these created? Is it something safe? How can we imagine that?
So we use our well-established patented PICVD technology to apply these inner coatings. And PICVD stands for plasma impulse chemical vapor deposition. It is a short patented technique, and its uniqueness is that it ensures a covalent bond between the coating and the glass substrate. And this differentiates it from other solutions that are used as barriers. A covalent bond means a bond on atomic basis, so the strongest chemical bond achievable. And the technology works like this. The vial itself is actually used as a vacuum chamber, and then HMDSO, so hexamethyl disiloxane gas is blown into the vial. And easily said, this gas is then split into its components. And what is achieved by the application of heat and microwave, based on how you adapt these parameters, so temperature, pressure, pulse, frequency, duration, different kind of coatings are achieved that bond to the glass substrate with different properties, fulfilling different purposes.
So really, you continue the chemistry masterclass. Thanks for the explanation. So I'd like to come to my last question, Diana. When should the customer think about the primary packaging?
So actually, a typical thing that we see, especially with smaller companies, is that they are, of course, focused on the efficacy of their drug, of their product. And thereby they oversee that the primary packaging is an integral part of safeguarding this efficacy. And you might not think, oh, okay, that only applies for the drug container interaction part, but I have to say, no, it applies for all kind of mentioned topics. We just recently had the case that a customer filled vials over nominal volume with a deep freezing process, so at minus 80, and he experienced breakage that put the clinical trial on risk. And with the right vial, this also could have been prohibited. So to summarize, it is crucial to consider the primary packaging at a very early stage.
Thank you, understood. So appreciate your time, Diana, and your explanations for us in this interesting talk, in which we try to guide the audience through the challenges of this bunch of next generation of drugs, of therapies, and the solutions that SCHOTT Pharma provides to keep all of them safe and very efficient and to be ready to reach very fast the patients all around the world. So thanks a lot and hope to talk to you very soon.
Bye-bye, Niels.
And thanks to the audience for listening and supporting us. Sorry. We really hope we've triggered good ideas and hope to meet you soon. Bye-bye.